Hi, I'm going to do a quick demonstration of how to use the IGV to view some of our structural variants from our mutation population. This is a mutant population of Panicum hollii that were irradiated with fast, which just means high energy neutrons. So this session has already been set up, but the way that we get it set up is you go to Preferences. There's a lot of things that you can change in here. I like to look at fairly large regions. And so for some of these settings, I will change it so that I am able to see larger size regions of sequences. So let's see, that is right here. So 30 kilobase is not terribly large when it comes to it. It really depends on your computer. I've done, oh geez, I think I've visualized about 5 megabase, and it would take a couple of minutes to render each time. I don't recommend getting that large, but you can certainly bump that up a little bit. So right now that's 300 kilobase. If I do that, that's 3000. So that's 3 megabase. That's probably a bit much. I'm just going to leave it at 300. I haven't really played a whole lot with the other settings, but I'm going to move forward. Yes. RNA. Same thing here. So for RNA, it's already set to 300 or above. Not that I'm looking at RNA terribly often, but I could. Cool. So finally, you need to set this up according to the data set that you're looking at. For my fast neutron population, my students have access to information in the write-up for this video on what to put into this URL, data registry, blanks. But that allows us to connect to the correct software server. So once you've done that, you do have to reset it. Obviously, I didn't change anything except for that viewing size, so I don't really have to reset it. But if you had just set it, you would then restart. So once I've done that, I would be able to load from a server. I have a login information for this particular server. Once I do that, I get access to this. Now, one thing that you may have needed to do if you've never logged into that server before, once you've got everything set up, this is me parsing JGI's multiple IGV servers. That's what those settings were doing. It was allowing me to tap into the list of available files. So I would have scrolled down. We're one of the newest data sets. So we were at the bottom. I guess they fixed that. Yeah, there we are. So you don't really want to download them all. It's a humongous amount of data. You would be waiting a long time and eventually probably run out of room. So there's several different data sets. These are just all of the DELI information. It's not terribly useful to visualize. Unfortunately, it's just too much data. This is the other type of variant analysis that has been done. These two combined is what's currently available on the variant browser for this population. I'm going to visualize the original lines. This is the mother of all of these. It's mostly inbred, so it represents them very well since there isn't much segregation going on since it's so inbred. And then I'm going to visualize a single sample. And this idea of kind of visualizing two at the same time is important because there is some weird mapping that just happens. So here I'm visualizing this mutant alongside the original genotype, so that offers a little bit of a control. It's going to take a minute. You're not going to see a whole lot even once it loads. There we go. So it's right now we're way too zoomed out. This is the entire genome. That's quite a bit larger than 300 kilobase. I'm going to go to a particular region. So I have a list of variants that I'm interested in. This particular variant is one in a plant that's turning red, and it's a knockout of a gene that breaks down beta carotene. So it's a really good candidate for being the culprit. And so I'm going to use that position information. Gonna go to chromosome 2. I believe it's, or one second, let me just zoom into a region. Couldn't remember what the syntax was. It's a dash to get the range. So I'm just gonna copy and paste and add a dash. And that seems to have got me to the right region. All right, so now I'm visualizing it. And you might, if I zoom out, let me zoom out a little bit. So that was a deletion event. And so this is one of this is the original parent, so it's not mutated. And here's the other one. So right off the bat, and let me right click on this. Oh, right click over here. I'm going to squish them. It just allows us to compact how it's viewed a little bit. Right away, I can kind of see the deletion.
Yeah.
There does seem to be a bad mapping region across this locus, though. So like, why is this so low, even though everything else is so high? Most likely, it's just a badly mapping region. The reads that belong there, the algorithm couldn't figure out where to put them. And so it's mostly not sequenced. However, that's clearly different from what's happening down here. Now, this isn't a very, like, you can see it, and that's cool, but it's actually not a terribly useful visualization. There are some colors being thrown around. And so if I go through my various settings, while right-clicking in a blank area, I can see that currently it is colored by the insert size and pair orientation. So these are what we call paired end reads. And so paired end reads, let me just pull up a diagram real quick. Yeah. Paired end reads. Yeah, that's a decent, that's a better picture. So paired end reads are when you sequence like a fragment of DNA. So you just randomly sheared or you maybe targetedly cut the DNA into different pieces. And then those pieces are read on a sequencer, but the sequencers can only read so much sequence. And so a strategy to get kind of more information is to read some amounts on one side, some amounts on the other side, and know how long that total piece should be. Because what that tells you is now, let's say you have 150 base pair reads, you have 300 total base pairs of sequence that have to align to a reference, but you can constrain how far apart they are. And what that tells you is that it reduces the amount of random alignments or inappropriate alignments that you could generate. And then this visualization is coloring by the size of that insert, because here and over here, we can see that some reads have a really large insert size. And that's hard to visualize. And so there's a good way to do this. So instead of just visualizing the actual alignments, I am going to view as pairs. And this is the real big trick for structural variants. At least it's really good for deletions. Because when I do that, all of a sudden, I see that those red marked sequences were marked red because they're linked across a deletion event. And so let me do that over here, view as pairs, and we don't even come close to seeing that pattern. I can then sort my sequences. I can sort them by insert size. However, grouping by reference concordance does a very similar thing while still showing kind of the orientation. And so what you'll find is it's not uncommon for mutations to be associated with a second mutation, especially big structural variants. So like I have seen a locus where there was a deletion on one side of the chromosome, and it looks like maybe an inversion on the other side. And so what I would see here is that there was two stacks with an inversion and with these deletion events. And so this is just a nice way of kind of organizing. All right, so there is some weird kind of alignments that occur up here. They're discordant. I think they just match. There's just got errors and stuff in them. But here there's an entirely new group of discordant sequences. And that's just because there was a deletion. But now, actually understanding the deletion better can be tricky. So you can do things like zoom in. This is a pretty large deletion, so it's kind of tricky to zoom into. I could zoom in over here. I could zoom in over here, try to see, are they consistently cutting off at a certain spot? And so if I'm trying to kind of validate that initial variant detection, I would look at that edge and say, all right, no, that makes sense. That's at position. Let's see, can I control Z that? No, I cannot. Dang it. It's good software. It's not perfect software by any means. So what I was trying to do was click slightly below where I clicked, drag in this open space, and zoom in on the region containing the mutation. Did I do it again? All right, well, it's because I'm using a tiny mouse and I'm trying to be fast about it. But that's the idea, is that you can kind of zoom in, see where the actual mutation occurred, and validate everything. Now, actually viewing the genome at this location and getting like gene information. All right, well, I'm going to stop doing this in front of everybody. Getting the gene information can be really useful. Cool. Was there any other questions that came up?
Just in terms of looking at other mutations, like, is that just a matter of playing around with it?
So inversions invite like where a translocations where the sequence has gone to another location. There are some tricks, but they're kind of niche and it's situation dependent. But essentially, IGV has the ability to split your screen into two different regions of the genome. And so you can kind of see where one read starts and then ends on a completely different chromosome. But it's only possible because we have this like very fancy way of aligning this type of sequence because we expected this to happen. We expected there to be these massive translocations, inversions. That's a very specific analysis. But yeah, it's a good question. Cool. Thank you. I'm going to stop this recording. So it's less weird. Bye.